Have you ever just stopped and wondered how your body builds anything? I mean, from the proteins that make up your hair to the enzymes digesting your lunch, it all has to come from a set of instructions. But how in the world does a cell read its own instruction manual? Well, the answer is this incredible process called DNA transcription. Think of it like a secret library system running inside you right this second.
Okay, so here's the big puzzle. In our cells, in complex cells anyway, the DNA, our entire genetic blueprint, is tucked away safely inside a little compartment called the nucleus. It's kind of like the world's most valuable book locked away in a library's special archives. So, if the instructions can't leave the library, how does any of the work get done in the factory of the cell, which is outside?
The cell, it turns out, is incredibly clever about this. It would never, ever risk taking its one-of-a-kind, totally irreplaceable master recipe book out of that protected room. No way. Instead, it uses a much safer, much more efficient system.
All right, so let's dive into that master blueprint. We're going to look at this problem the cell is facing and the absolutely brilliant solution it's come up with to read its own instructions without ever putting the original copy in danger.
And that solution is called DNA transcription. Officially, it's the process of copying information from a segment of DNA into a new mobile molecule. This molecule is called messenger RNA, or just mRNA for short. This is, without a doubt, the essential first step to turning a gene's instructions into actual action.
So to put it back in our library analogy, all transcription really is the cell making a quick photocopy of just one recipe, a single gene, from that master book. And this copy, the mRNA, it's cheap, it's disposable, it's literally designed to leave the library and be taken to the cellular kitchen, the ribosome, where the real building, the real cooking is going to happen.
And you know, this whole idea was a huge mystery until pretty recently. Back in the 1950s, Francis Crick was the one who first proposed this general flow of information. He called it the central dogma. But it wasn't until the 1960s that two scientists, François Jacob and Jacques Monod, actually figured out the mechanism. They're the ones who discovered how the cell made this genetic copy. And their work? Absolutely revolutionary. It turned a theory into a cornerstone of all modern biology.
Okay, so we get why the cell needs a copy. But how does this molecular photocopier actually work? Well, it turns out to be this beautifully organized three-step process. The whole thing is incredibly logical. First up, you have initiation. This is where a little molecular machine called RNA polymerase finds and clamps onto the start signal of a gene. Next is elongation. The polymerase just zips along the DNA strand, reading the code and building a matching RNA copy letter by letter. And finally, termination. It hits a stop signal, lets go of the DNA, and releases its brand new RNA photocopy out into the cell.
Now that middle step, elongation, is where the magic happens, and the machine follows a very simple rule. If it reads a G on the DNA, it adds a C to the RNA. If it reads a C, it adds a G. Simple enough. But here's the one little twist. If it reads an A on the DNA, it adds a U, that's uracil, to the RNA, not a T. Just remember AUGC, and you've basically cracked the code.
Now, that messenger RNA we just made, it might feel like the star of the show, but it definitely doesn't work alone. It's actually part of a whole crew of different RNA molecules, and each one has a very specific job to do. So let's meet the team. Of course, you've got mRNA. That's our message, the photocopy itself. Then there's tRNA, which acts like a whole fleet of delivery trucks, bringing the raw ingredients, these things called amino acids, to the construction site. The factory itself is actually built out of rRNA, which forms the ribosome. And in complex cells like ours, you even have snRNA, which acts like an editor, snipping out unnecessary bits of the message before it's sent off.
And you know, this process isn't a one-size-fits-all deal. In simple life, like bacteria, prokaryotes, it's a quick and dirty process that happens right out in the main cell fluid. But in complex life, like us, eukaryotes, it's way more elaborate. It happens inside that protected nucleus. It uses different polymerase machines for different jobs, and it involves that extra editing step we just talked about. Okay, let's zoom out and look at the big picture. We've seen how the copy gets made, but where does this one single step fit into the entire assembly line of life? It's time to connect the blueprint to the final product. And here it is. This is the central dogma in action. You can see the information flows from the permanent DNA blueprint to our flexible RNA copy that's made during transcription. That copy then gets read in a whole other process called translation, which is what actually builds the final functional protein. See, transcription is the critical bridge that connects the blueprint to the building phase.
And really, that's the bottom line. That's why this all matters so much. Transcription is the living link. It connects the static, unchanging archive of our genes with the dynamic, active, working machinery of the cell. Without this constant process of copying and sending out messages, the blueprint would just sit there, completely useless. Life, as we know it, just couldn't exist.
So this process is about as fundamental to life as it gets. But it does raise one last, really interesting question. We've been calling it a photocopier, right? And we all know what happens when a photocopier jams or makes a mistake. So what happens to us when the cell's secret scribe makes a typo? Well, that's a whole other story.